I think it is. It's really clear that a corner has been turned. I think largely in the last year, at least as far as a really broad base of participation goes. And that makes it a good time to try to think in a somewhat serious way about where we're headed. So this morning I'm going to try to step back from the level of detail that quite appropriately dominates meetings of this type and make a few suggestions about how, as a community, we should move forward to kind of meet the opportunities and really assume the responsibility that Rita Beery, for example, has put on our shoulders. Let's start really by making an obvious scientific point, because we should keep the unique characteristics of the science that we're doing central to the more tactical and even strategic aspects of trying to develop a fruitful marriage between kind of data-intensive molecular biology and clinical medicine. So the central scientific challenge is to try to understand the relationship between genotypes and phenotypes in humans. That's obvious. What I think does sometimes get lost is how unusual that scientific challenge is. The first Cold Spring Harbor meeting I came to was in the summer of 1977. I had a few hundred base pairs of DNA sequence, including what I believe was the first sequence of a mutant eukaryotic gene. I knew that this mutation caused the phenotype attributed to it because I was working in yeast and had at my command the tools of the model organism geneticist, inbred strains, experimental crosses, unequivocal evidence of causality. I enjoyed, I think it was Golson Lyon's comment yesterday, that we should be careful about using the term causality in a human context. And what he was touching there is the really tip of a large iceberg. It is essential that we understand as a community that the scientific challenge associated with understanding the relationship between genotype and phenotype in humans is entirely unique, not just to genetics, but to a large degree to science at large. We don't have any models for how to do this. Now, it's not that we haven't made any inroads. We know some things about the subject, and I wish actually I had some time to put my own favorite spin on this topic. I'll have to refer you to a review article just published in which I attempted to develop an evolutionary perspective toward this core problem. What is the genetic basis of human individuality? I'm going to have to leave that for another day because I want to stay focused on the future of this marriage between data-intensive biology and clinical medicine. So, 3 cautions. So my first caution is that I think we should change the name of this field. I think personalized medicine was perhaps a useful rubric under which to launch this activity, but it sends a misleading message, actually both to ourselves and to the broader community as to what the goal is. It's certainly true that there's much to be learned from the individual case. I enjoyed the discussion, for example, Barry Taylor about statistical blasphemy, and his talk and several others illustrated the power of the single case. But we need to attach our field firmly and irrevocably to evidence-based medicine. The main reasons that healthcare is better today, much better than it was when I was a child, has relatively little to do with molecular biology and even, for example, to advances in pharmacology. Really, at the core of this progress, I think any reading of the record of the last 50 years unequivocally kind of shows this point, is the adoption of evidence-based medicine as the central driver of progress. The idea of trying to figure out from first principles what is an appropriate way of managing a particular medical problem has failed spectacularly in the past and will fail in the future. And if we think that we know enough about cell biology, about physiology, about this and about that, to look at a single nucleotide variant in someone's genome and tailor their treatment based on first principles to that change, we're making a serious mistake. So, my proposal, which it comes from, at least to me, I'm not sure of the full intellectual history of this debate, but came to me from Steve Galley, the chairman of pathology at Stanford, is that we adopt the term precision medicine. Our goal is to improve, using molecular information, the precision with which we categorize patients and tailor treatment. But it has the virtue, and I'm going to come back to this because Galley's suggestion found its way into the title of what I think is an important National Research Council report on this subject that I'll discuss later. But precision medicine sets out the goal that really must be our goal. Is that we want to reduce the heterogeneity of the patient populations that are being dealt with in a particular way in the healthcare system. And I certainly am not trying to distance myself from the importance of dealing with extremely rare conditions, but the message there is that we need to expand our scope to very large populations so that even for rare cases, we will typically have groups, albeit in some instances fairly small groups, about which we can have a shared learning about appropriate medical management. So, I like, for example, Harold Varmus's anecdote on this topic. He is, at least I think, another convert to the precision medicine idea. At a workshop last June at the NCI, he just said a few introductory words that I found kind of moving, that his father was a general practitioner, and he said to the assembled cancer biologists that my father practiced personalized medicine. He knew the names of his patients. He visited them in their homes. What he couldn't do is use molecular knowledge of the underlying pathology in their cases to target treatment appropriately. And this is what we need to learn how to do, not just anecdotally in a few favorable cases that are sort of out at the edges of academic medicine, but in the mainstream clinic. And one of my principal cautions today is that our thinking kind of rise to the magnitude of what we're trying to do. This is an immense challenge. We're talking about $1 trillion industry just in the United States, a conservative one with a vast array of vested interests at every table assembled about how to move forward. Of course, it's a worldwide issue. Billions of people will be caught up in the web that we are just in the very earliest stages of weaving. So, having established the goal to lay the molecular foundations for precision medicine of the future, my second caution is to think a bit more carefully about the timescale that we're confronting and really the time dynamics. That is, how does something of this magnitude develop even if one is convinced, as I am, that the train has left the station. This is happening and will grow according to a dynamic that is difficult to foresee, but will involve spectacular growth. How does big change happen in society, particularly when it's driven by scientific and technical developments? So my thinking here, and really my only kind of special personal background that gives me any standing in this discussion, is my experience with the Human Genome Project. Following that Cold Spring Harbor meeting in the summer of 1977, I worked in pleasant obscurity for about 10 years during a phase which could be called the latent phase of the development of genomics. The dominant paradigm of the time was intensive application of molecular biological and recombinant DNA tools to the study of particularly the regulation of particular genes. And this was what dominated a Cold Spring Harbor symposium, a Gordon conference, papers in Nature, and so forth. But there were a few people laboring away at tasks which are still very familiar at this meeting. When you assemble shotgun data, what kind of problems do you run into? Well, you run into problems that are still familiar to anyone in this audience who has worked with current sequencing data. So that was 10 years in which I was pleasantly sheltered from Beltway politics and sort of involvement in sort of more policy aspects of science. That changed with the establishment of this National Research Council Committee, Mapping and Sequencing the Human Genome, which issued its report in 1988. It's chaired by Bruce Alberts and is often referred to as the Alberts Report. It has a strong prestige today in the Beltway because it's viewed as one of the relatively unusual instances in which a group of scientists sort of got together and got about right the delicate prophesizing function of these committees. Did not overstate what could be done in 1988, but did not understate what could be accomplished with a specialized effort directed at developing and deploying the technology throughout biology to sequence whole genomes and interpret the results. There's a principle that perhaps many of you have encountered in one context or the other, largely a Silicon Valley phrase that is attributed to Roy O'Mara, an American futurist, which is that in technical change, we overestimate the amount of change that's going to occur on a short time scale, let's say on the order of 5 years. And we underestimate the amount of change on longer time scales, let's say 10, 15, 20 years. And it really was that dynamic that we were struggling with in the Human Genome Project. Those of us closest to the ground, Shirley Tillman was the other junior member of this committee. At one coffee break, Shirley and I challenged the other committee members as to whether any of them had sequenced 1,000 base pairs of DNA with their own hands. Dave Botstein was the only claimant who, in addition to Shirley and me, who stepped up. But we disallowed his claim because it was based on the yeast URA3 gene. And I'd read this paper. The gene is 1,100 base pairs, and there were 7 authors on the paper. But those of us closest to the ground just knew that this wasn't going to happen in 5 years. It didn't matter how much money you spent. It didn't matter what you did. It just wasn't going to happen. And of course, throughout the development of the Human Genome Project, we always had to try to balance the let's just do it kind of argument. What we've got now is good enough. And the idea is that we can do a much better job if we solve this problem and that problem, and we're pretty confident we can do that. Compromise is always needed on these matters. So I'm going to sort of finish my cautionary comments by trying to address the really hard problem, which is how do I envision that compromise today? I'm certainly not arguing that we shouldn't move ahead with the worthwhile things that are being done, and we had many moving examples at this meeting. People's lives can be improved. Change is happening. But O'Mara's law reminds us that we do have time before this historical trajectory really accelerates and disseminates sort of beyond a phenomenon that a group like us can actually have much influence over. We have time not just to change the name of the field and along with it its public message, but to lay the groundwork for open-ended success. We need to develop an approach that will allow a virtuous cycle of scientific progress, improved clinical application, and then more of the same. And this is not a goal of the Human Genome Project type, where reasonably well-defined success could actually be achieved on a finite time scale, and then the field could move on. We're trying to build the groundwork here for this open-ended success, this kind of virtuous cycle. So, there are a lot of issues. I'm going to focus on one, which I think is at the core. And it's at the core because the human genotype-phenotype relationships are at the scientific core of what we're doing. And that's how we handle the data. The handling of the data for this project, issues surrounding the handling of the data kind of lay over this meeting like a thick layer of fog. And there were many suggestions, most of them relatively vague, about how to address this issue, but the recognition that the way we're handling the data now is not commensurate with our goals was ubiquitous. So, my third caution is that I believe the thinking on this topic is too conservative. It's easy to understand why it's too conservative. And for example, the ethics panel yesterday laid on the table a fairly familiar panoply of reasons that favor conservative thinking about handling the data. Problems everywhere. I have trouble even following the standard advice to call them challenges because they just sound like problems to me. So, I mentioned earlier that my enthusiasm for the phrase precision medicine came from my involvement with another National Research Council committee which issued its report last fall toward precision medicine, building a knowledge network for biomedical research and a new taxonomy of disease. So this committee was ably chaired by Sue Desmond-Hellman, the chancellor of UCSF, and kind of the key project manager, I believe, for the development of Herceptin at Genentech, and Charles Sawyers at Sloan Kettering, who played a critical role in the early Gleevec clinical trials. He's a CML oncologist. I served on this committee, and we took a first pass at this question. And I think there's much of value in this report. But the report operates at a relatively high level. It's a consensus document. Consensus always runs in counter to the need for more radical action. So here today, I'm going to exercise my academic freedom by proposing more radical action. Although it is entirely consistent, I believe, with the general principles articulated in this report. So, I think what we need is to envision and implement what perhaps could be called a GenBank for the 21st century. So, what are the characteristics of this database? It's going to be organized around the anonymized individual, not by the species or the locus or the chromosome, by the pathway or by the haplotype block. It's going to be organized by the anonymized individual, number 1, number 2, and I hope eventually number 10 million and counting. It's going to contain, obviously, molecular data, sequences and other types of molecular data that become, as time goes on, increasingly relevant to the clinical challenge. It's going to have long-term, that is, ongoing linkage to the electronic medical records of the individuals. And will include supplementary phenotypes as available. A number of speakers made the point quite correctly that you can't count on EMRs alone, but let's start somewhere. EMRs at least usually allow you to tell whether someone's still alive that you did a study on 10 years ago. I think, frankly, most of the molecular oncology literature that is flooding out should be quickly forgotten because we simply don't know what the outcomes were in these cases and have no way of finding out. If we're going to have an impact on mainstream medicine, a big impact, we're dealing with chronic diseases that progress slowly, that have complicated courses, treatments, environmental influences, social values are all going to change dramatically during the course of that disease. In the case of the individual, there is no other method than linkage to the ongoing activities of the healthcare enterprise that will allow the longitudinal character on the numbers of individuals that we're discussing. Obviously, many countries have healthcare systems better organized to this task than my own. And I want this to be an international effort from the outside. However, some of the ideas that I believe that we must not just consider but adopt, at least at the pilot scale level, are sufficiently radical that the United States actually may be fertile ground on which to try them. And I'm going to comment on how I envision this happening. So, organized by the anonymized individual, containing molecular data, long-term linkage to the EMRs, supplementary phenotypes as available, and full public access. One of my colleagues at the meeting gave me this button, free the data. I believe came out of the kind of pushback against the myriad model for handling genotype, phenotype information. We're going to need to free the data. So, why do we need to do this? Well, many reasons were articulated at this meeting. I've already commented on a couple of them. But let's just acknowledge that we not only don't have the information base, the scientific information base, to achieve the goals that I've outlined, but we don't have a system in place by which we're going to acquire that knowledge. Someone coined the phrase, the $1,000 genome, the $1 million analysis. This captures something that is real about our field, certainly at the moment, but is a deeply misleading metaphor. The problem with this metaphor is that it implies that Warren Buffett or Bill Gates could get his genome analyzed by paying enough bioinformaticians to comb over every base pair, whereas I couldn't, and someone even less well off than I am couldn't even think about it. This is not the problem. It's not even a significant chip of the problem. The problem is that we don't know how to analyze a genome in a way that provides any kind of broad guidance for the care of the typical patient. And our challenge going forward is to figure out how are we going to learn what we need to know to create a virtuous cycle in which this knowledge improves. It's going to need a collective solution, that's obvious. Numerous speakers have said that, in which patients, researchers, physicians, the whole system, our whole culture participates. I'd like to endorse an idea that I believe originated with Barbara Wold, who's worked for the last year rather selflessly on leave from Caltech as a sort of special advisor to Harold Varmus at the NCI. And against a lot of difficulties, which she can speak to herself, she has tried to establish the notion in the Beltway of the cancer information donor. I think this is an inspired kind of idea. It's like an organ donor. I checked on my Oregon driver's license that if they airlift me out of a wreck, they should at least kind of refrigerate my body and make whatever use of it they can. This is sort of built on this notion that I want to be a cancer information donor. I'd like to generalize it, medical information donor. And my hope is that 10 years from now, instead of these sort of free Huey type lapel pins, that we'll have more discreetly designed ones that say, I'm a medical information donor. We want to gradually change the culture so that people recognize that the real moochers in this system are not people who have government-subsidized health insurance. They're going to be people who expect state-of-the-art medical care but are not willing to share the information that has been gained by their own treatment. I believe this is an argument that we can sell to the public if we do it carefully. It's a good argument, and I think people will buy it. So, we need to do this. Why shouldn't we do it? Well, that would take too much time. We don't have that much time. Let me just share from my experiences in the Human Genome Project the two classes of arguments that we may expect. And I've encountered actually both of these arguments at this meeting as I set up trial balloons on this topic. So the first argument is that we're already doing it. It's going to happen anyway. The other argument, often from the same person in the next breath, absolutely impossible. It's a utopian, quixotic idea. And then they're back to already doing it. It's going to happen anyway. If you went to an ASHG meeting in, let's say, 1989, the more molecularly inclined human geneticists tended to say, we're already doing it. I sequenced 100 kb around one of the exons of the Huntington gene. And this is going on everywhere. It's all going to coalesce into a human genome sequence and so forth. No special effort is necessary. Well, this was abject nonsense and could easily be refuted by not just qualitative but quantitative arguments, not to mention technical ones. It, for example, set aside the issue that massive infrastructure, almost a complete reinvention of recombinant DNA techniques was needed to make this a practical goal, that we weren't just talking about staggering in Apollo program style, with some semblance of a human genome sequence, but changing the way biology is done to an information-driven science and so forth. So qualitative, quantitative arguments abounded, and that's why people needed the fallback position that it's impossible. Well, we're certainly not doing what I described. There is not one patient in the world today where I can go from this meeting, log on, and examine their genome sequence, their electronic medical record, and the associated phenotypic information, and so forth. I mean public access. I don't want committees. This is like GenBank. I can go down to my local high school and mentor a bright kid who wants to do a science fair project by analyzing sequences in GenBank. And 10 years from now, I want to be able to go down and I'll have a lot more takers if I can offer them a science fair project where they dig into this database and learn some new things about health, disease, life. So yesterday's ethics panel laid out many of the reasons why this is impossible, and that major compromises are necessary. And I don't want to discount the strength of the points. We all need to educate ourselves rather carefully about what is a nuanced and sophisticated literature on these subjects. But I did inject from the floor the notion that I thought rather missing from the panel yesterday is a principle that must be our main guide, and that is to avoid genetic exceptionalism unless it is absolutely necessary. Invoking exceptional ethical features of genetic data should be a last resort, invoked in particular situations, not a first resort. The first resort should be to rely on a far more robust, far more experience-based, and far more realistic bioethical framework that has simply grown up around the sensitive information about individuals that we're discussing. I believe that with these principles, we can move forward. So how to move forward? This idea is too radical for government or for large institutions. George Church got that right about the Personal Genome Project, which was pioneering in sort of breaking down the fear about public identification of sequences and so forth, but was not designed to meet the task that I'm laying down today. So eventually, this venture will need public support, but it's a fool's errand, I assure you, to go to Bethesda kind of with a white paper on this topic. We need to get together an ad hoc coalition. The Open Software Foundation is a sort of a rough model here. That is, do something pretty complicated that normally would require a different sort of organization, different sorts of funding. Put together this coalition, researchers, the genomics industry, pharma, healthcare payers, foundations, wealthy individuals who find this argument convincing, raise some money. We only need a few $1 million to get started. This is a trivial sum compared to almost any relevant metric in this field. Enroll the patients and post the database on the web. Start with about 100 patients, a very modest number, but I don't want the money to be going into the production activity of enrolling more and more patients. I want it to go into building the core infrastructure. It's not going to be very fancy, but figure out how to solve the innumerable problems that we will encounter and get this database up on the web. The goal really being to show that it can be done, to refute the argument that it is impossible. Then the bet is that by these mechanisms we can lead government, large institutions, and the public toward a future that I think can support the ambitions that have, in a very moving way, been at the center of our discussion at this meeting. So I had 3 cautions. Let's get the name right, and really what I mean there, let's get the message right. We have to sort of think exponentially, not linearly. This is an alternate statement of Amara's law. We're still in the rather flat phase of an exponential curve and tend to be thinking incrementally about what we can do next year and next year and next year. We've got to be preparing for when this curve starts to turn up, and we will lose control at that stage to much broader social forces. At this point, people in this audience can really influence what this future looks like. And the one that I've outlined is by no means the inevitable, indeed at the moment would be considered a long-shot candidate for what this future is going to look like. But let's give it a try. So that was the last caution, is that half measures, leaving it to someone else, is going to accelerate the process of control moving away from scientists who've devoted their lives, their careers, and their emotional commitments to this venture, and it will lie in the hands of our political system, our economic system, our large institutional framework, and we will be sniping from the edges where at the moment we own this. So, that's my message. Let's resolve to build on what has been a very exciting experience. Try to do some of the good in the world that Rita Berry so eloquently charged us to do. And have some confidence that we can do this. Thank you.
I'm sure that will have stimulated some questions amongst many of us.
Yeah, I agree 100% with everything you just said. I think that's an amazing talk, an amazing suggestion. Just one thing is, the Personal Genome Project, of which I am a participant with George Church. I mean, he did try to get Google Health and try to get electronic medical records. I mean, it's already, so there's that that's going on. Why do you think that can't be transformed into the project that you're describing?
So I'm open to allies. I've known George since he was a graduate student, and my perception is that the Personal Genome Project has really accomplished its goals, and did so actually rather against a lot of obstacles. It has taken away the taboo, where we went from only Jim and Craig and a few people, that are accustomed to being in the public eye, being willing to have their genomes out there, and made it, a more acceptable idea. I don't see, although I'm certainly happy to talk, with George or with anyone. I don't see the kind of what amounts to the political operation there that is going to be required to get this done. I think the initiative needs to be moved a little more into the establishment, not too far, but a little more. We need the support of CEOs and various people that are not going to join an outright insurrection, and so we need to kind of file off some of the edges.
Thanks so much, Maynard. And in complete agreement with the problem and the landscape, there is perhaps another analogy for moving forward. Rather than needing the gen bank, we might need the pregnancy test. So where the thing goes from something that's quite technical and deep in the hands of the limited scope of the clinical care community becomes a ubiquitous test you can get everywhere and becomes more commoditised. And that in itself may raise the tide and transform the landscape. So I guess my question is, if we can't achieve this, the utopia you project of the gen bank with all the phenotype data in it that's freely publicly accessible, are there any other pathways that can lead us to a more optimistic future rather than simply have the American College of Pathology lock this up?
Too much into the establishment. I'm looking for a middle zone. But the American College of Pathology is going too far into the establishment. But anyway, I think I understand. Yeah, right. I think I understand your point that home pregnancy tests, HIV tests, and so forth kind of empowered the people and disempowered kind of an establishment that had a very paternalistic way of kind of dealing with people. I'm certainly open to suggestion, but I don't see how we get there, because the problem with, say, the 23andMe products is kind of obvious. It's the one that I stated. I like the rhetoric of this company, but the information base isn't there. So you can sell to the public, the idea that if you get your genome sequenced, there's all this information in it, and we don't have the way of capturing that information. So that's why I'm advocating selling to the public the idea that we can do this together, that we can offer some things already, but not much. The consumer genomics industry has been forced, just by business constraints, to argue that there's real good value here right now. And I think we need a different message, because I don't think the value is that good in the drugstores.
So I wonder what you think about the idea of, with regard to this database, having essentially an iTunes for genomes. And could you speak more to why this has to be done in the public arena versus the private sector? Because iTunes did revolutionize the music industry, and you do provide incentive for the user and the depositor providing their data and making it open, provided the fee is small enough per use.
Yeah, I think models like that are interesting, and I don't know a huge amount about them, but they did. iTunes, for example, did have the characteristic that it had what is now the most highly valued, the highest capitalization company in the world kind of behind it. That is, yes, it was sort of democratization in one sense, but there were a lot of decisions that were made by relatively modest groups of people about how to do this, and they countered what a lot of the free the data, free the tunes kind of community thought that they would be inevitably defeated by peer-to-peer sharing and so forth. But so all I'm really saying is that this will need some structure. It's got to be in this sort of intermediate zone between the American College of Pathology and the National Research Council on the one hand, and really just kind of Cold Spring Harbor bar talk on the other. It's got to have a little structure to it. Look what the Open Software Foundation did. I mean, Linux is now running a sizable fraction of the servers in the world, and is a bulletproof operating system, and it was developed by outside of the establishment. But there was quite a bit of structure. We need some structure, because there are a lot of decisions that need to be made, and we need to be able to gain the confidence, of establishment people that this is at the minimum a worthwhile experiment that's being done by serious people that they can have confidence in, as opposed to a bunch of kids in their dorm rooms trying to revolutionize the way intellectual property is handled or mishandled.
Maynard, so I'm taking chair's prerogative.
Yes.
So if you think of yourself coming back in 20 years' time and the issues that we've discussed the last few days, which of them do you think you'll be saying we grossly overestimated and which we grossly underestimated?
Wow. It's Yogi Berra's comment about that it's hard to make predictions, especially about the future. There probably will be applications of this technology, and I'd be reluctant to guess which ones they'll be that we're not anticipating, that become the killer applications and that develop the public momentum. And I can't say kind of what they'll be, but that's what usually happens. Is that there's a lot going on in the Amara's Law framework during this sort of more lag phase. And there are a lot of Darwinian things going on, and then boom, it really goes critical. I mean, look, the whole IT industry missed email as the kind of defining application of the internet. They just didn't get it, and boom, and it was there. So something like that will probably happen, but I'm not going to guess what it is.
Well, thank you very much, Maynard. So our next speaker